Good morning, one and all. I am Dr. Bindu PP, Assistant Professor, Department of Statistics, Government Arts and Science College, Calicut. Today, I am going to discuss application of Bayesian methods for analysis of clinical trial data. Now, we will look what is clinical trial. A clinical trial is basically an experiment designed to evaluate the beneficial and adverse effects of a new medical treatment or intervention. Clinical trials are performed with human subjects to test new drugs or combinations of drugs or new approaches or new procedures to improve disease diagnosis or patient's quality of life. We know that clinical trial has 4 phases. Phases represents the stages. For example, the development of a new drug which requires early dose finding and toxicity data in human. Indications of potential activity, comparison with the standard to determine the efficacy, and then post-marketing trials. These are required to market a new drug or interventions. Now we will discuss what is phase 1 trial. Phase 1 trial is the earliest type of studies that are carried out in human beings. That is the first trial in human being. They are done using small samples, around 20 to 40 of healthy subjects, and this is done to investigate pharmacodynamics, pharmacokinetics, tolerance and toxicity of the drug. After successfully completing the phase 1 trial, it will then move to phase 2 trial. Phase 2 is carried out in patients, usually to find the best dose of drug and to investigate safety. Access the response rate or survival to find an effective dose using sample of 30 to 100 subjects. These are exploratory trials aimed at discovering if a compound can be useful clinical results. After successfully completing the phase 2 trial, it will move to the phase 3. Phase 3 is a comparative trial aimed at conclusively demonstrating the efficacy. They are sometimes called confirmatory trials. Here the sample size usually 250 to 4000 human beings. Carried out in patients, usually to find useful effect of the given drug with, compared to the standard drug available in the market, and also to find the best dose of drug and to investigate safety and advantages of the new drug with the or the existing drug. After successfully completing the phase 3 trial, it will move to the phase 4 trial. Phase 4 trials are generally considered as post-registration trial. After phase 3, the product can be registration, and phase 4 is a massive trial, and it is called post-registration trial. That is trials of products that already have been marketing authorization. Phase 4 trials are carried out for the products that have marketing or the FDA approval for marketing. Pharmaceutical companies used to carry out studies that were solely for marketing purpose. Bayesian methodology of the design of clinical trials is to incorporate relevant information into the design process. There are a lot of statistical methods used in each phases of the clinical trial, and Bayesian methods are very useful in analysis of clinical trial, and the analysis and interpretation of the for the analysis and interpretation, several authors used Bayesian methods. In Bayesian methods, available information pertinent to the trial is summarized into using a prior distribution. Final trial data is combined with the prior or updated prior to obtain the posterior distribution. Bayesian estimates of treatment effects and corresponding credibility intervals can be calculated. We know that Bayes' rule tells us how to combine the prior distribution with the likelihood to find the posterior. Bayesian inference overcomes several limitations of frequentist statistics. It has a lot of applications in many areas. It permits inductive inference by reporting the clinically relevant probabilities for specified treatment effects. Application of Bayesian methods to design and analyze clinical trials are done by various authors, Friedman et al. 1988, and recently FDA Center for Devices has encouraged hierarchical Bayesian statistical approaches. Bayesian hierarchical modeling techniques have some advantages over classical methods for the analysis of cluster randomized trial. Also, Bayesian methodology is useful in meta-analysis of multiple treatment comparison. Janssen et al. 2008. Optimal Bayesian feasible dose escalation for cancer phase trials. Sack et al. 1998. Also, Bayesian methods are used in sample size determination, and also in clinical trials, sample size determination plays an important role in the design aspects of the study in many fields, especially in planning of the clinical trials. Applicable to equivalence trials, non-inferiority and superiority trials. In superiority trials, establish that a new experimental treatment is more superior than an existing treatment. In frequentist approach, a given test of the null hypothesis of no difference between two treatments. We specify alpha and power 1 minus, with alpha, level of significance alpha, and power 1 minus beta, and we can determine the sample size using this formula: sigma square into z1 minus beta minus z alpha by 2 whole square by theta a, where theta a is value of theta under h1. What does z represent? The normal, standard normal, table values represent the table values. Because of the uncertainty involved in the specification of unknown variants, Bayesian concepts are better for sample size determination. Bayesian approach has lot of advantage over the frequentist approach. Possible approaches have been uh, classified as hybrid classical Bayesian, decision theoretic Bayesian, and proper Bayesian. You can refer several papers on this. Frequentist Bayesian hybrid approach consider the predictive probability of achieving the primary study goal based on the available data, aims to control type 1 error. Second uses the length of posterior credible interval as the sample size criterion. Because of the uncertainty involved in the specification of unknown variants, Bayesian approaches are better for sample size determination. Also, the Bayesian approach chooses sample size to maximize an investigator or selected utility function or minimize a corresponding loss function. We also consider developing a new two-stage fully Bayesian sample size estimation procedure for phase 2 trial design to test the efficacy difference between a drug and a placebo for a binary endpoint. Beta prior to the treatment effect and sample size estimation is based on the posterior. We are developing the algorithm to applied on hypothetical clinical data or bioassay criteria. We used our program for the simulation studies and to analyze the data set. And in this study, in conclusion, there are numerous areas with the clinical design and analysis where we can use Bayesian approach. Bayesian approach has a lot of application in the analysis of design and analysis of clinical trial data. This includes planning of clinical trial, sample size calculation, subgroup analysis, meta-analysis, interim analysis, etc. FDA 2006 guidance for the use of Bayesian statistics in medical device clinical trials. For reference, you can look FDA 2006 for the use of Bayesian application of Bayesian trials and guidelines for the use of Bayesian statistics in clinical trial analysis. These are the references. Thank you.